November 1905. The air inside the modest biological laboratory at Bryn Mawr College is a strange cocktail of chemicals and life. It smells of formaldehyde used to preserve specimens, of the dry, earthy scent of mealworm bran, and of the faint, metallic tang of the gas lamp that casts a focused, golden circle of light onto a brass microscope. Within that circle of light, a world is being revealed. This is Nettie Stevens' modest Bryn Mawr lab, and for months this has been her entire universe. At 44 years old, an age when many of her male contemporaries were already tenured professors with established reputations, Stevens was a research associate, a position of precarious prestige fueled not by institutional support but by sheer, unyielding intellectual curiosity. Her focus is absolute. She is dissecting the reproductive organs of the common mealworm beetle, a creature most would dismiss as a pest. To Stevens, it holds a fundamental secret of existence. The process is an exercise in extreme patience, a microscopic surgery performed with fine needles and a steady hand. She teases apart the delicate tissues, fixes them to glass slides, and applies a series of chemical stains designed to make the invisible visible. Inside the nucleus of each cell lies the genetic material, which, during cell division, condenses into the thread-like structures we call chromosomes. The scientific community at the turn of the 20th century was in a feverish debate about heredity. Gregor Mendel's work on inheritance in pea plants had just been rediscovered, but the physical mechanism, the how, was a mystery. What cellular structure carried these hereditary traits from parent to offspring? Many suspected the chromosomes, but the evidence was circumstantial, the theories abstract. And the most perplexing question of all was the determination of sex. Was it nutrition? Temperature? The age of the parents? The theories were as numerous as they were baseless. Stevens believed the answer wasn't in the environment, but locked inside the cell itself. Slide after slide passes under her lens, hundreds of them, each one prepared with the same meticulous care. She draws what she sees in a notebook, her pencil strokes precise and confident. She is building a data set, an unassailable wall of empirical evidence. And then it happens. Not in a single flash of insight, but as the culmination of 1,000 observations clicking into place. A pattern, undeniable and revolutionary. In the cells taken from female beetles, she consistently counts 20 large chromosomes. They appear as 10 matched pairs. But in the cells from male beetles, the picture is different. There are 19 large chromosomes and one conspicuously, consistently tiny one. It's a stark asymmetry in a system that was presumed to be symmetrical. She examines the sperm cells. Half of them carry 10 large chromosomes, the other half carry 9 large and that one tiny, distinct outlier. The egg cells, in contrast, are uniform. They all contain 10 large chromosomes. The implication is staggering, a biological thunderclap. If an egg is fertilized by a sperm carrying the 10 large chromosomes, the resulting embryo has 20 large chromosomes. It becomes female. If, however, it's fertilized by a sperm carrying the 9 large and 1 small chromosome, the embryo has 19 large and 1 small. It becomes male. The small chromosome isn't an accident or a deformity. It is the determinant. It is the switch. She had just identified the XX and XY chromosomes, the biological basis of sex determination. The large chromosome, present in both, she would term X. The small, male-specific one, she designated Y. It wasn't about diet or temperature. It was a simple, elegant chromosomal lottery. Her resulting paper, Studies in Spermatogenesis with a special reference to the accessory chromosome, published that same month, is a masterpiece of scientific clarity. It doesn't speculate, it demonstrates. It lays bare a fundamental mechanism of life with the cold, hard evidence of observation. Yet, science is not a pure meritocracy. It is a human institution, rife with rivalries, ambition, and systemic bias. Just as Stevens was finalizing her work, another, more famous cytologist was treading similar ground. Edmund Beecher Wilson, a distinguished professor at the prestigious Columbia University, was a giant in the field. He had access to more resources, a team of graduate students, and a powerful network of colleagues. He, too, was studying insect chromosomes and came to a similar conclusion, publishing his findings almost simultaneously with Stevens. His theory was slightly different at first. He proposed that the male had one less chromosome than the female, rather than a smaller, distinct Y chromosome. But his position and influence were immense. He represented the scientific establishment, the world of Ivy League professorships and institutional power. Stevens was an outsider, a woman from a women's college working largely alone. The world was presented with two papers describing the same monumental discovery. One came from the center of academic power, the other from its periphery. One from a man, the other from a woman. Whose name would history remember? Following that groundbreaking paper, the scientific world should have been lit ablaze with the name Nettie Stevens. Her discovery was not a minor clarification, it was a foundational pillar for the new science of genetics. Her methodology was a model of scientific rigor. While other researchers, including Wilson, had observed odd or accessory chromosomes before, they had often struggled to connect them directly and consistently to sex. Stevens, through her exhaustive work on was the first to provide a clear, unambiguous, and evidence-based demonstration of the XX/XY system. She didn't just suggest a correlation, she proved a mechanism. She showed how the segregation of these specific chromosomes during sperm formation and their subsequent combination with an egg determined the sex of the offspring with mathematical certainty. This was the tangible physical evidence that linked Mendel's abstract factors of inheritance to concrete structures within the cell. It was, in short, the key that would unlock the century of genetics to come. But the currency of science is credit, and credit flows along established channels of power. As the nascent field of genetics began to coalesce in the years that followed, a subtle but relentless process of erasure began. Textbooks and review articles started to tell a simplified, more convenient story. E.B. Wilson, with his professorship at Columbia and his influential network, was an easy figurehead. He was a known quantity, a respected leader in the field. His name began to appear first and then often alone. The narrative began to shift. It became Wilson's discovery. Stevens, if she was mentioned at all, was relegated to a secondary role, her contribution framed as a mere confirmation of Wilson's more insightful work. We can see this happening in real time in the archives. In early editions of major genetics textbooks, she might receive a sentence or a footnote. By the 1920s and 30s, as the citation landscape solidified, her name often vanished completely. This wasn't a conspiracy orchestrated in a smoke-filled room. It was something far more insidious, the quiet, unquestioned operation of systemic bias. It was the assumption that the man from the elite institution must, of course, be the true genius, and the woman from the small college a diligent but secondary contributor. Let's be clear about the injustice here. Wilson himself, in his initial papers, did credit Stevens's work, but the system around him did the erasing for him. Thomas Hunt Morgan, another Columbia professor and future Nobel laureate, was initially a skeptic of the chromosome theory of inheritance. When he finally came around, his influential Flyroom group built their Nobel-winning work directly on the foundation Stevens had laid. Yet, in their historical accounts and lectures, it was their colleague Wilson who was consistently lauded as the pioneer of sex chromosome research. The Matthew effect, as sociologist Robert K. Merton would later call it, was in full force. To those who have, more will be given. Credit, funding, and recognition accumulate for those already recognized, while those on the margins are stripped of their contributions. The very structure of academia, the way papers are cited, the way histories are written, the way reputations are built, work to sideline her. Stevens didn't have a legion of graduate students to carry her name forward and embed it in the next generation of textbooks. She had her data, her papers, and her quiet, unyielding dedication. In the face of the academic establishment, it wasn't enough. This erasure had profound, tangible consequences. Despite a career that included over 40 published papers and a discovery that reshaped biology, Nettie Stevens never achieved the professional security of her male peers. She was promoted from research associate to associate in experimental morphology at Bryn Mawr, but she was never granted a full professorship. She spent her career applying for grants and fighting for lab space, always having to reprove her worth. The recognition that should have guaranteed her a stable, well-funded career was instead redirected, fueling the prestige and resources of others. We are left with a haunting question, a question that probes the very heart of scientific history. Who benefited from this narrative shift? Whose career was propelled by the very insight Stevens had painstakingly uncovered alone in her lab? The answer is an entire system that found it easier to credit a familiar patriarch than to acknowledge the revolutionary genius of a woman who dared to solve one of nature's greatest puzzles. Despite the institutional pushback and the systemic forces attempting to diminish her role, Nettie Stevens' work was too fundamental to be ignored, even if its author was. Her discovery became the bedrock upon which the entire edifice of modern genetics was built. The XXXY system wasn't just an interesting quirk of mealworm beetles, it was a near-universal principle of animal life, including our own. The impact rippled outwards, transforming medicine, agriculture, and our basic understanding of what it means to be alive. That direct line from her microscope slide to our 21st century world is the most powerful testament to her legacy. Think of the diagnosis and understanding of sex-linked genetic disorders. Conditions like hemophilia, red-green color blindness, and Duchenne muscular dystrophy are all caused by recessive genes on the X chromosome. Because males have only one X chromosome, a single faulty copy is enough to cause the condition. Females with two X chromosomes are often protected carriers. This entire field of medical genetics, which has saved countless lives and allowed for informed family planning, rests on the fundamental principle that Stevens demonstrated in 1905. Every genetic counselor who explains inheritance patterns, every doctor who diagnoses a sex-linked condition, is using knowledge that flows directly from her work. Her discovery is the conceptual root of technology she could never have imagined. The medical practice of karyotyping, creating a visual profile of a person's chromosomes to detect abnormalities like Down syndrome or Turner syndrome, is a direct technological extension of her meticulous cell-by-cell observations. The very idea that you could look at a cell's nucleus and find a predictable physical blueprint for an organism's sex and potential genetic vulnerabilities began with her. Her work provided the crucial, tangible link between the abstract patterns of inheritance and the physical reality of the chromosomes, paving the way for the gene mapping projects and genomic medicine that define our era. From forensics to evolutionary biology, her identification of a distinct, heritable physical marker of sex created a new paradigm. It is, without exaggeration, part of the genetic blueprint of life itself. For decades, this foundational contribution remained a story told only in whispers, a footnote in the annals of science. But history is not static. It is a living discipline, constantly being re-examined and corrected. In recent years, thanks to the tireless work of historians of science, particularly women like Marilyn Bailey Ogilvy and Stephen G. Brush, the true story has been painstakingly reconstructed from archival letters, lab notes, and a critical rereading of the original scientific papers. They have peeled back the layers of accumulated bias and revealed the undeniable primacy and clarity of Stevens's research. This is the act of restoration. Institutions are slowly beginning to right the historical wrong. Bryn Mawr College now proudly claims her as one of its most important scientific figures. Biographies are being written and articles are being published that place her correctly at the center of the story, not on its periphery. Today, as we work to build a more inclusive and equitable scientific community, her story serves as both a cautionary tale and an inspiration. It is a stark reminder that genius has no gender and that we must remain vigilant against the subtle biases that seek to diminish it. Efforts to ensure Nettie Stevens' invaluable contributions are recognized are about more than just correcting an old error. They are about reaffirming our commitment to a future where credit is awarded based on the brilliance of the discovery, not the identity of the discoverer. Her legacy is finally being restored, her name brought out of the shadows. But the question remains, how many more essential truths, discovered by marginalized thinkers, lie buried beneath layers of historical injustice waiting to be resurrected?